Hello everyone, this is Smaya Kattimani. I'm graduate student in Applied Cognition and Neuroscience. Today I'm talking about the research paper Decoding Native Cortical Representations for Flexations and Extensions at Upper Limb Joints using Electrocorticography. The research background in this paper mainly focuses on the usage of a technique called electrocorticography, which is used to decode the cortical representations of the motor cortex in our brain. In this paper, we can see that it is used to decode the cortical representation of flexation and extension movements at the upper limb joints. In this paper, it is also investigated the use of brain-machine interfaces, which actually helps in controlling the limb movements, specifically focusing on discriminating between flexation and extension of different limbs joints using the electrocorticography recording from the sensory motor cortex. BMI research has focused on modeling endpoint reaching tasks for neurally driven prosthetic arms, primarily using a Cartesian coordinate center approach. The important research question through this paper was is it possible to restore function to patients with upper limb paralysis or limb loss? When it was systematically investigated for the first time, to check the flexation and extension movement involving fingers, wrist, and elbow, they could classify the electrocorticography signals recorded from the sensory cortex at various spatial scales. They also found that the power in high gamma bands of electrocorticography signals provide enough information for them to classify the different movements using the linear classifier. And also, movements were able to be distinguished at the macro electrocorticography and HD electrocorticography scales than the micro electrocorticography scales. Then the results suggest that the neural representation of movements of the joints were maybe separable enough using the electrode spread over the sensory motor cortex to provide the BMI user to control over individuals. In research methods, there were 4 individuals with epilepsy who underwent invasive monitoring with electrocortico arrays were selected, and they were selected based on their clinical indication for epilepsy surgery, and when they agreed to participate in the study, they were instructed to do the random flexation and extension motion of their upper limb, which includes finger, wrist, and elbows. And this experimental testing was conducted for the period of 7 to 14 days, and the major target implant region was sensory motor cortex, from where the neural calculations were analyzed. In this slide, we get to see the brain map reconstruction for the patient 1, 2, 3, and 4. And for the patient 3, 2 microelectrodes are located on the sensory motor cortex, which are used for the analysis. And the central sulcus is outlined in black. During the monitoring period, participants were asked to perform specific hand and wrist movements related to flexation and extension of upper limb joints. Neural signals were recorded from the primary motor cortex and sensory motor cortex, and the recorded neural signals were subjected to various signal processing techniques. Time frequency analysis was used to analyze the spectral characteristics of the signals in different frequency bands. The decoded cortical representations were determined by applying a linear regression model. The study also included a signal coherence analysis to examine the degree of synchronization between the neural signals recorded from the different electrode sites. For the decoder evaluation, statistical analysis was conducted to assess the significance of the decoding accuracy. This included the calculation of p-values and the use of appropriate tests to determine the significance of result. In this slide, we can see the plot which is showing the accuracy of classifying 6 moments, that is function of time, for patient 1, 2, 3, and 4. The blue trace represents the average time-dependent classification accuracy for 10-fold cross-validation, and the black horizontal trace represents average chance accuracy for 1000 shuffles with respect to movement onset, black vertical line. The blue shade represents the standard deviation of classification accuracies across the 10 folds. The red shaded region represents where the decoding accuracy was significantly higher than chance. Result, we see that the study underscores the importance of electrocorticography grid coverage in capturing widespread neural activities. In patient 3, it is demonstrated with high decoding accuracy across the multiple joints along the arrays of the sensory motor cortex. In patient 4, it is exhibited with decoding accuracies above chances before movement onsets. And it is also seen how individual joint flexation and extension movement could potentially accommodate movements in object-centered references frame. These applications include assisting patients with upper limb amputations through robotic prosthetics and helping individuals with upper limb paralysis through functional electrical stimulation or control of exoskeletons. The paper suggests methods to improve decoding performance, including the hierarchical linear classifiers and the two-step decoder that combines movement classification and regression steps. These techniques have the potential to enhance the BMI control and broaden the range of activities achievable by the users. In summary, the paper highlights the potential and challenges of using electrocorticography signal for decoding upper limb movements and discusses their applications in clinical settings, particularly for individuals with limb disabilities. The several key highlights regarding the use of electrocorticography for decoding upper limb movements are: it shows modality for decoding neural signals associated with the limb control. It may reduce the need for extensive user training compared to the other methods, and optimizing electrocorticography grid coverage is crucial for accurate decoding. Sensory feedback plays a significant role even in patients with altered sensory input, and pre-movement neural activity offer potential for enhanced control. Electrocorticography can enable object-centered limb control benefiting various tasks. Clinical applications include assisting amputees and individuals with limb paralysis. Advanced decoding techniques can further improve accuracy and versatility. Overall, electrocorticography holds promise for enhancing the limb control and has significant clinical potential.